turn them into cell-based therapies, one of the ways that we can do that is transduction. Let's take a closer look at transduction. In fact, one of the most common ways to deliver genes into cells is to use viruses to do it for you. In fact, viruses already have the machinery needed to transmit the genetic information inside the viral capsid or shell into the target cell of interest, and then get that target cell to produce the proteins for which that gene encodes. The basic mechanism of transduction is actually just endocytosis. When the virus, which is sometimes called a particle or a vector, comes into contact with the cell membrane pinches off around it and forms what's called an endosome. And so you can see that pinching off allows the virus to fully enter the cytoplasm. And then one of two things happens. In the case of retroviruses that carry RNA, the RNA is actually released into the cytoplasm, where it's converted into DNA by reverse transcription. And then that DNA makes its way into the DNA of the host genome. So what we mean here is that the DNA from the virus becomes part of the DNA of the target cell and can replicate whatever is coded for in that DNA. In the case of an adeno-associated virus, or AAV, the viral particle escapes the endosome entirely, and then that whole particle releases its DNA into the nucleus. The difference here is that DNA does not integrate into the host genome, and it remains tied up in a little circle called an episome. Either way, the cell begins producing proteins based on the genetic information that came from the viral vector. So, for example, in Unit 1, we discussed genetically engineered immune cells like CAR T cells. And so you can imagine that the genetic information for a specific cell surface receptor, a protein, is expressed and coded in. And that's how you get a chimeric antigen receptor, or CAR, to be expressed on the surface of a T cell. Now, here are the two of the most commonly used viral particles used to deliver genes into cells for cell therapy. A retrovirus is about 100 nanometers across, and it has a viral capsid or envelope around RNA. So retroviruses carry RNA. The R's go together, so that's easy to remember. But the AAV, the adeno-associated virus, is much smaller. It's only about 20 nanometers across. It doesn't have that envelope, and inside the AAV is ssDNA, or single-stranded DNA. So one of the obvious comparisons here is that the retrovirus is much larger than the adeno-associated virus, and therefore you can fit more genetic information into a retrovirus. Now, there are two important considerations when transducing cells for cell therapy manufacturing. The first is the virus-to-cell ratio, and we refer to this as the multiplicity of infection. This is an empirical parameter. We choose it in our process development, and then we adopt it. So this virus-to-cell ratio, or MOI, could be 1-to-1, and in this case I have one virus for every one target cell. But if it's, say, 1/2-to-1, that means I have fewer viruses than target cells, here 1-to-2, one virus to every 2 target cells. Or if I have an MOI of 10-to-1, I have an excess of viral particles to target cells, here 10 viral particles for every 1 target cell. The second consideration for transducing cells for cell therapy manufacturing is the functional titer, and this is information that you can obtain from the viral vector manufacturer, or you would measure if you yourself are the viral vector manufacturer. The key here is that the number of viral particles per milliliter of fluid volume is not the same as the number of transducing units per mil of fluid volume. Let's think about why that might be. First of all, you know the number of particles per mil, but you do not know how many of those particles actually have DNA or RNA inside of them. So you might have empty vector. You know the number of particles per mil, but you don't know how many of those particles actually have an intact capsid that's going to be able to interact with the target cell and infect it. And finally, you know the number of particles per mil, but virus biology is broad and variable and not completely understood. And so some viruses are more infectious than others. So for all these reasons, knowing the number of viral particles per mil of solution is not sufficient or equivalent to knowing the number of transducing units per mil that you need to introduce into your target cell population. Now, once you've gone through that step, how do you actually measure whether that transduction was effective? There's two ways that we do this in the manufacturing process. The first is that we compute the transduction efficiency. The second is that we compute the vector copy number. And there's important reasons for doing this related to both dosage and safety. So in terms of transduction efficiency, let's say I add about 10 viral particles to about 4 target cells. The transduction efficiency doesn't tell me that ratio or multiplicity of infection. It actually tells me how many of those target cells were transduced successfully. In this case, only 1 out of 4 expressed that gene product of interest, that protein on the surface. And so my transduction efficiency is 25%, 1 out of 4. It's possible, theoretically, that I could have applied those viral particles to the same number of cells and ended up with 100% transduction efficiency. In that case, all 4 of the cells were able to express the gene product of interest. Now, there are several things that affect the transduction efficiency that help you get from that range of low transduction efficiency to high transduction efficiency. The first is the raw material quality. Now, this could be the quality of the viral vector itself, for example, related to the number of times you've had to freeze and thaw it in your processing steps. And it could be related to the quality of the target cells, such as the age of the patient or the number of times that that cell batch has been frozen and thawed. But the transduction efficiency is also highly modified by the cell culture parameters while you're exposing these cells to the viral vector. That could vary with the temperature, the pH, the composition of the media, or even the time of incubation. And so all of these steps can drive your transduction efficiency up or down. But in practice, transduction is never 100%. It can range depending on all of these variables and from cell product to cell product, but a common transduction efficiency through current processes is 25 to 30%. So if you don't have 100% transduction efficiency every time, how does that affect the final formulation and the dosage? Well, here we think about two things. We have to make up a rule for how many of the cells, what percentage of the cells are expressing the gene product of interest to say that production was a step and that transduction was efficient. So we could say, let's set our threshold at 25%. And that will actually decide whether or not we release that product to the patient because we had sufficiently high transduction efficiency for a threshold that we set for ourselves. But remember that the dosage to the patient requires that they receive cells that actually express the gene products of interest, not all those other cells that aren't expressing the gene products of interest. And so the dosage is actually set by the number of transduced cells. This means that right now, if you don't separate the transduced cells from the non-transduced cells, and that's common practice, you have to add more total cells to the patient if you have a lower transduction efficiency threshold. Now, thinking about how this all relates to safety, we can think about the transduction efficiency as quantified by vector copy number. And vector copy number simply means the number of copies of a gene that came from the viral vector and got inserted into the host genome. If there's 1 copy of that gene, the vector copy number is 1. If there's 4 copies of that gene, the vector copy number is 4. But having a higher vector copy number is not necessarily a good thing. In fact, because of some of the risks that we'll get into, it's typical to keep the vector copy number to lower than 5. So what are those risks? One of the risks is called insertional mutagenesis. Now think about a target useful medicine for the patient. That target cell might have a gene within it that isn't active. That gene is turned off and not expressed, but the gene is an oncogene. And that means if it were turned on, when you introduce the new genetic material from a retrovirus, that genetic material could be far from the oncogene and have absolutely no effect on that oncogene, or it could insert close to the oncogene and end up unintentionally and inadvertently turning that oncogene on. And now the cells that you intended to provide as a safe medicine have become cancerous. So this is a very serious concern, and it's one of the reasons why we limit the vector copy number so that we don't insert that gene that comes from the viral vector in too many places, because that uncontrolled proliferation is a form of cancer. So another risk of viral transduction is what we call the replication competent vector problem. Remember that these vectors were designed to deliver genetic information into the target cell to produce a good outcome, and definitely not designed to produce more of the viruses themselves, because that would turn our target cells into virus factories and would create a risk of infection. Even though these viral vectors were designed to limit that replication competence, we still check it every time, because just like you could turn an oncogene from off to on, you could have homologous recombination that could actually turn a replication incompetent vector into a replication competent vector in this context. Now, in early primate models, this was observed to occur, and that was generation 1 of these vectors. We've learned from that over the years of engineering the vectors to avoid this challenge or at least mitigate the risk, and now we're at generation 3 and 4. And so we would take more recombinant combination events to produce this risk. However, it's still important to check it every time because safety is of utmost importance in these cell therapy products. So what have we learned? First, viral vectors can be used for transduction and therefore insert new genetic material into target cells and make them into useful medicines. The key parameters that control this process are the multiplicity of infection, that's the ratio between the number of viruses to the number of cells, and the functional titer, the transducing units per mil of the viral vector itself. The key measures of transduction effectiveness include the transduction efficiency, that percentage of cells that express the gene products of interest, and the vector copy number. How many times do we get that gene inserted into the cell's genome? The viral transduction has a number of risks, and those risks that we've covered include insertional mutagenesis, which can lead to cancer, and replication competent vector, which could produce viruses unintentionally. So while these risks are considerable, they can be offset by the potential benefit to the patient by creating new kinds of medicine that impart new functions to cells as cell therapy.